right, welcome to the MAPS Canada webinar series. My name is Shannon Smedella, and I'm your host for the series. We are extremely excited to have you join us for our 5th episode of the webinar series. Throughout the next few months, we have an amazing selection of speakers, luminaries, scientists, doctors, and visionaries lined up just for you. We've gathered some of the best in the field, 9 additional speakers to be exact, to share important information and their take on the psychedelic renaissance. We are in the middle of the psychedelic renaissance, with research projects and decriminalization proposals popping up across North America, and with MDMA on the verge of approval as a treatment for PTSD, psychedelic therapy is on track to become the next major breakthrough in medical healthcare. MAPS Canada, the Multidisciplinary Association for Psychedelic Studies, has supported studies to develop psychedelics into safe and legal medical treatments since its formation in 2008. Currently, MAPS Canada is funding 2 separate studies on MDMA and cognitive processing therapy as a treatment for PTSD. Additionally, plans to fund studies that identify psychedelic treatments for eating disorders and others that involve ibogaine are in the works. To conduct and publish scientific research and education supporting the beneficial use of psychedelic medicines, MAPS Canada relies on public donations and funding initiatives. The funds raised from this webinar series will go to fund this research. For more information on MAPS Canada, please see the website at mapscanada.org. So we are going to start today with a talk from our guest, Paul Stamets. After that, we will start our question and answer period. So make sure to vote up your questions, which you can see at the bottom of the screen, as we will prioritize those. And without any further ado, I would like to introduce to you someone that you already know very well, Paul Stamets. Paul Stamets is a speaker, author, mycologist, medical researcher, and entrepreneur. Is considered an intellectual and industry leader in fungi, habitat, medicinal use, and production. He lectures extensively to deepen the understanding and respect for the organisms that literally exist under every footstep taken on this path of life. Paul is the author of 6 books, including Mycelium Running, How Mushrooms Can Help Save the World, Growing Gourmet and Medicinal Mushrooms, and Psilocybin Mushrooms of the World. He has discovered and named numerous new species of psilocybin mushrooms. Paul has been awarded more than 40 patents, with several patent applications in queue for unexpected activity of psilocybin analogues stacked with other substances. He has received numerous awards, including Invention Ambassador for the American Association for the Advancement of Science, the National Mycologist Award from the North American Mycological Association, and the Gordon Tina Wasson Award from the Mycological Society of America. His work has entered into the mainstream of popular culture in the new Star Trek Discovery series on CBS. The science officer is portrayed as an Astromycologist, a Lieutenant Paul Stamets. Paul's work with mycelium is a central theme of the series. Today, here is what we will be discussing. Psilocybin is one molecule of several that can cause altered states of consciousness. The focus has been on these active molecules. Is there activity of other molecules that co-occur and are related to psilocybin that do not alter consciousness, but have a demonstrable positive effect on brain health? Paul will discuss his research that contributes to the science of consciousness. Psilocybin mushrooms have been used for thousands of years. Psilocybin is a molecule that has been used for a few dozen. The reductionist view of medicine has been effective in discovering a molecular structure for treating bacterial infections, for instance. But the single molecule medicines available for psychotherapists have failed to show the same level of positive outcome. Where and when can the use of natural form of psilocybin mushrooms be appropriate for psychotherapy and cognitive health? How do you standardize a natural product? Could it be approved by federal health agencies? Does a patent like the one Compass has cross over to interfere with the use of natural products for therapeutic use? These are just some of the questions that will be answered in this episode. So please help me welcome Paul Stamets.
Hello. Hi, everybody. And I heard I got a promotion. Now I'm a lieutenant commander astromycologist, Paul Stamets, on the next Star Trek. So I gave them a lot of ideas on psilocybin too, but I'm not sure if they have the courage to use these ideas. We will see. But honored to be here. Thank you, Shannon. So, well, I have been studying psilocybin mushrooms all my life. I am excited to be here, a part of MAPS. I'm a large supporter of MAPS because of one primary individual, Mark Hayden. A big shout out to you, brother. Mark is a kindred spirit and soul, and he's helped shepherd MAPS Canada along with many other people. But I want to acknowledge Mark for his friendship, his leadership, and for having the vision to carry MAPS forward in the Canadian ecosystem. I'm talking to you from a remote island in British Columbia. I came up here, I have a cabin up here, and I came up here expecting to be here for 7 days, and 3 months later, I'm still here. In fact, I want to stay here. It's batshit crazy in the United States, and it's really normal up here. So it's nice to be in a country where science is respected, and the politicians actually seem to be behaving like adults in the room, not so much so in our country. So anyhow, a lot of Americans feel that way. I will say that if Trudeau opens the border, there could be a mass exodus of Americans coming to Canada. So watch out, brace for impact. So I'm going to cover a lot of territory, a lot of history. There's a lot of questions that I still have that have not yet been answered. But I like challenging authority with respect. And I like to authenticate everything I say. And when I don't know, I will tell you I don't know. But when you're challenging conventional wisdom, whether it's in therapeutic drugs for helping improve the emotional state of mind, your psychological state, whether it's challenging some of the conventional wisdom of traditional Chinese medicine, no matter what it is, us disruptors, people who ask these critical questions and who have new information, it's really common to have opposition. I welcome that opposition. And so I want to jump into my slides, and Shannon, thank you so much for that kind introduction. And I'm a Luddite, folks, so I will probably screw up here, but maybe not. Good God, it worked. So psilocybin mushrooms in culture and consciousness. So I will like to first show you, Mark Hayden actually first presented a slide similar to this. And I worked with Mark and I added more institutions, but just to put this in perspective, these are the university institutions currently engaged in clinical studies on psilocybin and psychedelics. So Harvard Medical School, Stanford, University of Pennsylvania, UCLA, Imperial College. This is enormous number of institutions studying psychedelics, but let's focus more now on just psilocybin. These are the institutions and academic centers currently involved in psilocybin research. Yale University, Stanford, et cetera, Mount Sinai. So bear in mind that before you get a clinical study approved, you have to go through IRB, Institutional Review Board examination by critical thinkers, experts in the field of medicine, to see if the preliminary evidence justifies a clinical study. Is it addressing an important need that other medicines do not currently address fully? Is it safe? Are the individuals conducting these studies competent enough to carry it through? Are the institutions, do they have the resources to be able to complete these studies? So it's a pretty high hurdle for many of these researchers to cross over, and indeed they have. I will note the US government, the US Department of Veteran Affairs is also very keen, specifically on the use of psychedelic medicine for overcoming PTSD. Our good friend Rick Doblin and Leanna Gilby are two of the individuals who've been spearheading this, also with a great team of other collaborators. So a shout out to MAPS United States and to Rick Doblin. So what's happening elsewhere? Well, in Europe, also the wave of interest in using psilocybin is sweeping across Europe. So this is really important because my best supporters I found to be my skeptics. Everyone is naturally skeptical. You have questions. When you're informed, you can make a more informed decision and judgment. But until you go up the learning curve, you're faced with the inherent biases or prejudices that everyone has. And that's just normal. That's what humans do. But there is such a consortium, a chorus of academic institutions all on the same page, recognizing the efficacy and the power of psychedelic medicine in a time critical, especially in the field of psychiatry, where so many of these drugs that have been approved have deleterious side effects that don't really lead to a quote unquote cure. It may lead to a partial recovery. It may dampen and suppress some receptors that are misfiring or overreacting. But it's amazing to me, since I've been involved in this since the early 1970s, to see and to be alive today, to see the preponderance of academic institutions seriously picking up this subject for clinical research. So this is now I know a number of other researchers out there have used a slide. And when I first saw the slide, I was as amazed as many of you, I'm sure, are. This is just a range of toxicity of harm to users and harm to others, like your friends, your family. And I grew up with an alcoholic father, and when he would get drunk, he would get angry. Now, I admit to drinking beer, but I drink a beer, I get happy. Well, that's really interesting. Different drugs, or even the same drug, affects people differently. So we're not all the same. But nevertheless, looking at a large meta-analysis, the danger of alcohol is very well established. And the danger of alcohol to people in proximity to you also has tremendous collateral damage to those people. When you look at the scale, you see mushrooms and psilocybin mushrooms basically on the extreme end of non-toxicity, and it's very, very few instances of collateral damage to anybody in close proximity. So mushrooms basically are very safe compared to the other drugs that are of note. I want to bring your attention to IP34. This is a Oregon Psilocybin Service initiative by Sherry Vesey and Tom Eckert, who are the chief petitioners. The Psy 2020 movement is a culmination of about 4 or 5 years since it was first conceived. And on Monday, June 29th, the Oregon Psilocybin Initiative has a major press conference. I would encourage everyone hearing this to spread the word and to tune in to their press announcement. The petition is for essentially the therapeutic use of psilocybin mushrooms for the citizens in the state of Oregon. So this is the first statewide initiative on the ballot, which will be on the ballot in early November of this year. So, excuse me, a big shout out also to David Bronner and David Bronner's group. I mean, David walks his talk. He is one of the most courageous individuals I've ever met in my life. He puts his money where his mouth is, literally. And Dr. Bronner's soap and products also carry a very important message on the legalization and support of psychedelic science. And so I would be remiss in not also acknowledging David Bronner and his team. He has a wonderful family of associates and employees that are also central players at Burning Man. And I've been in their camp at Burning Man now for about a half a dozen years. So now I populated a website called mushroomreferences.com. It's totally unbranded. I'm a victim of Google Scholar alerts. I get way too many of them. And basically, what I've done is I've curated, along with one of my employees, Renee Davis, we populate this website, what we think are some of the most significant references on the medicinal properties of mushrooms, including psilocybin mushrooms. So if we have the abstracts and we have links then to the original articles, should they be covered by copyright. But in mushroomreferences.com, you can see the enormous amounts of information on the psilocybin research that's been ongoing. You can see lots of information on showing the validation of mushroom mycelium. And the mushroom mycelium produces the enormous numbers of enzymes and extracellular metabolites that are constantly in crosstalk with the ecosystem. In over hundreds of millions of years, they've developed exquisite suites of compounds that allow the mycelium to navigate through otherwise hostile environments. So please check out mushroomreferences.com. And we update it usually about every 2 to 4 weeks with now another download of articles which we assemble. It's got a good search engine. It's got categories. You can type in any keywords and try to find references that are of interest to you. So the mushroom life cycle is something I just want to cover very quickly. This is very rudimentary, but basically, with the majority of mushrooms, 2 spores have to come together. They're haploid. They have half the complement of the chromosomes necessary to produce fertile offspring. So you basically, it's sort of like male, female, but they're mostly called mating pairs because there's more than just two sexes. There can be literally up to 23,000 sexual mating pairs with some mushrooms. But generally speaking, there's sort of 8 to 16 mating pairs in many of them. And if they are compatible, they come together, they fuse the uninucleate, mononucleate, 1 nucleus per cell in the mycelium. When the 2 mating strands of hyphae come together, they fuse, they become binucleate. Now they have 2 nuclei per cell. And now the mycelium races much faster. So when they're a monokaryon versus a dikaryon means they have 2 nuclei. Once they've mated, the mycelium tends to be a lot more aggressive. So the mycelium then grows, and it grows for weeks, months, years. And then after literally trillions of cell divisions, then in a very short window, typically often only 5 to 7 days, a mushroom is formed. And they suddenly pop out of the ground. But the mycelium has been there all along. And so the mushrooms pop up very, very quickly, and they are nutritionally dense. They attract mycovores, animals that eat mushrooms. And so actually the mushrooms don't have a very good immune system. They're designed to be eaten, they're designed to be discovered, and they are designed to rot. But the mycelium that gets to that stage in the mushroom life cycle has to navigate through a very microbial hostile environment. And that's why we're so excited about mycelium. It has this huge armamentarium of defenses that allows it, being a single cell wall thick, to navigate through hundreds of millions of organisms literally per gram, in many cases, to be able to survive, to create a mycelial mat sufficiently large enough to reproduce. So many people say a mushroom is to mycelium like an apple tree is to the roots of the apple tree. Yeah, that's true. Some people dispute that mycelium is part of a mushroom. Well, that would be like to say that the roots of an apple tree is not part of an apple tree. I mean, it's just nonsensical logic. A mushroom generates mycelium, and a mycelium generates mushrooms. So it's as simple as that. There's Psilocybe cubensis growing out over 5 days. As it grows out, it diverges. Every time it forks and diverges, there are new endpoints. And along that peripheral margin is a site of epigenesis. This is where the feeding mycelium, as these tentacles of mycelium are reaching out, if they encounter like a new insect, a new source of food potentially, even a new toxin, at that intersection, at that margin, at that interface, if epigenetically the mycelium can code for a new protein, a new enzyme that can unlock the nutrients in this newly found food, what happens? Well, it then surges in its growth, and that new enzymatic sequence that then becomes genetically encoded. And so then it becomes resident within the body intellect of the mycelium. So epigenesis is the expression genomically of a new gene sequence in response to an external environmental stimulus. So this is why the network-like design of mycelium is so profound. And it's more profound than most people realize. Now, my friend Nick Reed from England generated these. Patrick Hickey and Nick Reed worked together. Patrick Hickey gets the primary credit. And these are dark field microscopy images showing the streaming of nuclei through the mycelial networks. And prior to Patrick Hickey and Nick Reed making these, we didn't know that these nuclei would stream as bundles. And they call them comet nuclei, euphemistically. And that's extraordinary because the mycelium, if you went back and saw that again, it doesn't all stream in the same direction. About 90% of it does, but some of it actually goes backward against a cytoplasmic flow. And we think, hypothetically, is those bundles that are going backwards that have now encoded new information. And then the transference of the genetic new skill set is transferred back to the mother mycelium. The largest organism in the world is a mycelium mat in Eastern Oregon. I hired an airplane. I went up there twice. And this is a mycelium mat that is 2,200 acres in size. Now we know that's the size of the mycelium because it's from a honey mushroom that kills the forest, Armillaria astoi. There's also Armillaria gallica and solidipes, but this one is an Armillaria astoi. These are honey mushrooms. Most of you have seen them, but it's a forest parasite. It kills the trees. And so what happens when the trees get killed in these mountaintops, then they get baked in the sun. They no longer bring up water. So they become ashen gray and they are a huge fire hazard. So then they come into these forests that have died and they cut the trees down in order to limit, forest fires, of course. But that margin you see is the margin of the mycelium that is identified that's killed the trees. And doing about 24 samples across this 2,200 acres, they determined that indeed it's the same organism. So the largest organism in the world is a mycelium mat, 2,200 acres in size, and it's one cell wall thick. Think of that. You have 5 or 6 skin layers that protect you from the infection. The mycelium has one. Any of you who are biologists or microbiologists out there know this well. They're about CFUs, colony-forming units. You have healthy garden soil. You scoop up a gram of soil, you'll have several million colony-forming units, basically several million microorganisms. And in a cubic inch of soil, you can have more than 8 miles of mycelium. So think of the ability of the mycelium to be so pervasive, to be so well integrated into the soil microsphere, and yet be able to be in competition with literally hundreds of millions of microbes that are trying to consume it. And yet it achieves the largest mass of any organism in the world. How does it do this? Because epigenetically, it has coded an enormous suite of host defense molecules that allow it to compete in the natural world to achieve these huge masses. So the mycelium will grow out vegetatively as a network-like design in response to primarily 4 stimuli. After it grows out, you have introduction of water. We all know that, rain. When you have rain, you have evaporative cooling, so you have a drop in temperature. That's the second one. Third is that when the ground gets wet, the mycelium surges from underground up to the surface and then exhales carbon dioxide, inhales oxygen. So that's the third stimulus. And the fourth one that's quite surprising is light. Almost 98% of the mushroom species out there that have been cultivated require some degree of light exposure. So those 4 stimuli cause mushrooms to form from the mushroom mycelium. And the mushroom then forms these little baby primordia, and the primordia then very quickly enlarge into a discernible mushroom. So the primordium in most species are called deterministic, and that is that the cap and the gills and the stem tissue, all has been coated already in that small form. So if you took a razor blade and you cut that little mushroom like that, a cleft, the adult that would grow up, even a mushroom 6 to 12 inches in diameter, would have a giant cleft in it. And so, so they are at this stage of development, they're very, very sensitive. And they're also extremely sensitive to blue light. So in many of these psilocybin mushroom species, especially with the ligniculus, the wood chip growing psilocybin mushroom species, will not produce psilocybin or psilocin until it's exposed to light. And sometimes even not after exposed to light. But the absence of blue light it pretty much limits, if not prevents, the production of psilocybin and psilocin. So I started to explore how many primates consumed mushrooms. And I found this article that came out of Australia. And the Golgi monkey in Bolivia consumes more than 12 times its body mass per year in mushrooms. So I started to look, well, how many other primates do this? And it turns out 22 primates, 23 when you include humans, we're primates, have a discernible habits of being able to identify edible from poisonous mushrooms and consume them. So it's a major part of our food chain. So that's really interesting, because that speaks to a long ancestry of mushroom identification knowledge that not only humans have, but other primates have as well. So over 2 million years ago, our ancestors' brain suddenly enlarged. Homo sapiens appeared only 200,000 years ago. That's relatively recently. And so Homo erectus, came to Europe. Neanderthals migrated from Africa into Europe. 2 migrations of Homo sapiens. One first migration pretty much failed. Another migration around 40,000 years ago was successful. But about 200,000 years ago, the Homo sapien brain evolved and basically doubled or tripled in size from our primate ancestors. So that's really interesting, because within 4,000 years of contact in Europe, Neanderthals became extinct. We mixed our genes with Neanderthals. Yes, Homo sapiens had sex with Neanderthals. And so if you do a 23andMe test, I did mine, I'm 3.25% Neanderthal. I'm kind of proud of that. So we all have some Neanderthal in us. I think that's just about 95% accurate statement. But the brain also enlarged at a time of radical climate change. And so the enlarged brain putatively give the ability cognitively to come up with better solutions to survive an ever-changing and challenging climate. So this is my friend Louis Schwartzberg commissioned this. Let's see if I can get it to start again. Let's go back. And there we go. And this speaks to the stoned ape hypothesis that Dennis McKenna and Terence McKenna first postulated. The idea is our primate ancestors came out of the jungles, from being tree dwellers, and across the savannas, hungry, watching for animals. They would follow scat. They'd find these very bodacious mushrooms. They'd ingest them, and being hungry, 20 minutes later, lift off, a sudden psychedelic experience. Now you're hungry, you're going to share it with your loved ones, your children. And so these small clans are very likely to have shared these psychedelic experiences together. Now the question becomes, okay, can we prove this? This may be an unprovable hypothesis. They called it a theory. I disagree. Hypothesis is speculative, not necessarily borne out and tested factually. And a theory is basically a hypothesis that has been tested by facts. So I call it a hypothesis. But let's just examine, circumstantially what is going on here. So if you're a hunter, you're looking for footprints, you're looking for scat, you're looking for dung. The herd that you're tracking is over the horizon. So you're looking for dung, you're looking for scat, you're hungry. Look at the size of the mushrooms growing out of the scat. They invite you to eat them. You're hungry. They look clean, they're pure. So how many times will this happen? Not once, not twice, but millions upon millions upon millions of times over millions of years. This is why I think Dennis and Terence McKenna are onto something. That's a fundamental truth in the evolution of humans. And now we have the evidence that psilocybin mushrooms and some of the psilocybin analogues cause neurogenesis. So I think they were really way ahead of their time. This will remain and hotly disputed by paleontologists, but I think that science is well learned. You should be careful about being skeptical of ideas that can be so profound, because Copernicus, Galileo, etc., also shared in having the skepticism levied against them. And now, look at their place in history. So it's not a theory. It's a hypothesis in my mind. Psilocybin degrades into psilocin, it binds into serotonin receptors, stimulates neurogenesis, improving visual acuity and hearing. As I mentioned, it's a hypothesis, not a theory. So we go forward now, and now this is the earliest evidence that we believe of psilocybin mushrooms. Now this is the original cave art, and then it was redrawn by Kat Harrison, and then my friend Jonathan Meter. But it is true to form. So the image you see now is true to form of the cave art 7,000 years ago in the Sicilian Anjar Plateau in northern Algeria, now overtaken by climate change of the Sahara Desert. The Sicilian Anjar Plateau is known as the Plateau of Running Rivers. So at that time, it was a deciduous forests were prolific, and an artist is clearly trying to communicate their excitement about mushrooms. And mushrooms, because they rot so quickly, a traditional method in Mesoamerica and elsewhere in the world is to store magic mushrooms in honey. That may well lead to psychoactive meads. And actually mushrooms were banned in 1516 in the Bavarian Beer Purity Act, where henbane mushrooms and other ingredients were banned under pressure of the Catholic Church to remove psychoactive substances from mead because they would lead to this religious and intoxicating state. We could spend hours on this. So again, there's two images showing that they're true to form. Obviously, the artist is trying to communicate mushrooms and bees are paired together. And elsewhere in the cave are these running figures, and I find these particularly to be profound. Are those spores or are those ideas? But there's a communication from the head of the mushroom to the head of these running figures who, and this gait of dancing or running is, I think, signifies excitement. So another example of this. All of this is open to interpretation. We can't talk to these people. They don't have a written record per se, but they had pictographs and petroglyphs. And from these pictographs and petroglyphs, we are gaining some knowledge. So at the time of the origination of the myths in Greek culture, Persephone is receiving a mushroom from Demeter, her mother. And in the fall, this speaks to the Eleusinian mysteries and early European culture. For more than 1,000, some people say for up to 1,500 to 2,000 years, the Eleusinian mysteries were a sacred ritual using fungal concoctions where more than 1,000 Greek citizens once in a lifetime traveled about a dozen or more miles, heckled along the way, in order to receive the sacred rites that were held in secrecy by the priesthood. Sophocles, Aristotle, and Plato all partook of the Eleusinian mysteries. And then with the advance of Christianity, another example, monotheism, suppressing polytheism, history is written by the victors, and the religion that dominates then suppresses the indigenous wisdom and other religions that are looked upon as being a threat to the prevailing monotheistic culture. In Mesoamerica, these mushroom stones have been found About 200 of them are known in the world. And these were found in the Pacific slopes of Guatemala. And the mushroom stones went to about 600, 500 AD. Now, 1,000 years later, the conquistadors came in, but these mushroom stones were still being revered. And by a decree of the Catholic Church, all effigies that were threatening Christianity were to be destroyed. So these were apparently destroyed and smashed by the thousands. They have turned up in fields of farmers who plowed them, and they find them buried. They were buried, we think, because of secrecy, because the diseases that were brought over by the Europeans, the indigenous shamans had no treatment against smallpox, syphilis, potentially tuberculosis, and other diseases. And so the shamans' medicines did not work. So many of those who converted to Catholicism were promised everlasting life. That was a new concept. And so obviously they would start snitching on their neighbors because the diseases running rampant were obviously, blame somebody else rather than blame yourself. And so these mushroom stones are very few and rare. I am the temporary custodian of 28 of these mushroom stones. They've come to me throughout my life. I fully intend to return them to the Mayan culture. I'm trying to find a Mayan recipient who has the wherewithal to host these. It is in my will that these are returned. I have sought out several recipients, and I'm still trying to interview and find the ones who would be the proper ones. The reason being is I've been warned by several groups associated with the Guatemalan government, don't give back to the Guatemalan government, that should be sold off in the art trade again. And because these mushroom stones have value in the antiquities market, I think it's important that they're kept together. I feel like I have a Mayan village of elders, of ancestors. Every morning when I walk out of my bedroom, this is what I see. Every evening when I go into my bedroom, this is what I see. I feel the calling of experts in the field of mycology through the eons of time. We're all part of the sacred lineage, and it's intercontinental, it's intercultural. I think anyone who's become bee mushroomed, we all share in this love of what mushrooms can do for us. So I need to speed this up a little bit. Many of you know about Life magazine, 1957, or Gordon Wasson, Maria Sabina, is the first field guide, basically delivered to millions of Americans in the middle of the Cold War on magic mushrooms. I was mentored by four individuals: Michael Bugh, Alexander Smith, Daniel Stuntz, and Kit Skates. Alexander Smith and Daniel Stuntz and Kit Skates have now passed on. Michael Bugh was a professor at the Evergreen State College with whom I worked. And Michael received a Drug Enforcement Administration license because of several articles he published that could accurately determine psilocybin in psilocybin mushrooms. At the time, the scientific literature was not accurate, and the DEA was using inaccurate methodologies for analytical determination of psilocybin. So Michael was called as an expert witness to defend individuals who had picked mushrooms, and some of them actually were not psilocybin mushrooms. It was indeed a flawed analytical technique. He published an accurate one, and that sort of led to his way of getting a DEA license, which covered much of my work. Now, this is all the more significant because when I walked, basically, I was a logger hippie up in the Cascades. I was self-taught. I spent enormous amounts of time at the University of Washington Library with Daniel Stuntz. And when I came out of the woods, this is what I looked like, which I had a lot more hair back then, let me put it that way. And that was my first cook of Psilocybe cubensis in that little pressure cooker. So I'd like to set the history straight on a few things. Baeocystin is an analog to psilocybin, and it was named after Psilocybe baeocystis. And this is really a misnomer, and I'm convinced of this, because the reports came out of early reports in 1962. It was a few years earlier than that. 6 individuals. There were 2 adults and 4 children, 4 years, 6 and 9 years, on 3 separate occasions, ate some mushrooms that appeared in their backyard. And this is an event in Kelso, Washington. And they were then hospitalized. And some of them ran fevers, GI upsets, convulsions. One child actually died. And so they went back. One child actually went back 4 or 5 days later and grazed in the yard and ate some more. More than likely, it was a mixed collection, potentially Galerana autumnalis, a deadly poisonous mushroom. Some of the symptomology seems similar. But in looking for the potential toxin, the specimens were sent to Alexander Smith from the stomach of one of the individuals who died. And a single specimen was collected, and it was grown in culture from fragments of the mushroom. And the mycelial spawn then produced several mushrooms. Now, there's only a few of us who have ever tried to culture Psilocybe baeocystis. Psilocybe cyanescens is very luxurious white mycelium like that of cubensis, rhizomorphic growth. Psilocybe baeocystis is a very thin mycelium. It is unlike any Psilocybe mycelium I've ever seen. It dies out. It is highly unlikely that the Psilocybe baeocystis mycelium grew in culture to produce fruit bodies. I don't know anyone, even to this day, has ever been able to produce this in vitro. So I believe what they did is had collected Psilocybe cyanescens, and they misidentified this. I talked to Alexander Smith about this. The spores of baeocystis are very different than that of cyanescens. I brought that to his attention, and he agreed. And he also agreed that he may have misidentified these mushrooms. That happens, folks. Even the best experts in every field make mistakes. And so Alex, when I confronted him with this information and my skepticism, he did not disavow it. He did not say I was wrong. He said, you may be right. He had a vague memory of receiving, stomach washings basically and isolating out cultures. Alexander Smith published a monograph on the genus Psilocybe, so he was a psilocybin mushroom expert. The 1970s, the Great Psilocybin Mushroom Species Expedition in 1977. I'm second to the right. There's Dr. Gaston Guzman, who wrote a monograph on the genus Psilocybe. We started a series of mushroom conferences. This is the first one that we did on psychoactive mushrooms. And we did these, and this was one in Florence, Oregon. There's Jonathan Ott on the right, Jeremy Bigwood, Gaston Guzman from Mexico, Dale Leslie, myself, Jim Jacobs, and Stephen Pollock. So a series of mushroom conferences occurred, and then a proliferation of mushroom books on psilocybin mushrooms in particular began to be published. So in the 1970s, there was a wave of interest in psilocybin mushrooms because psilocybin mushrooms were being collected by young people in the field, Psilocybe semilanceata, and then in wood chips they used for landscaping around buildings. With the advent of wood chips and landscaping with bark, beauty bark and wood chips, massive numbers of psilocybin mushrooms, heretofore not seen, suddenly started appearing around universities and institutions and around courthouses and police stations. And to this day, around Google headquarters and Apple headquarters, the psilocybin mushrooms grow in the beauty bark. So they have this strange affection to following humans that are making important decisions. So Michael Bugh, mentor, Jonathan Ott, Jeremy Bigwood, and myself, between us, I think we've written about 17 books. And Mike is a great teacher and mentor who I dearly love and respect. Mike has a wonderful, kind heart, and he never would shame his students. Some professors are alpha, and they like to have one-upmanship, and they're highly competitive. And Michael Bugh is just epitomized one of the gentlest, nicest professorial personalities that I've ever seen. I went on to publish 4 new species: azurescens, linoformans, Cynofibulosa, and Psilocybe wiley, named after my friend Andrew Weil. And then Psilocybe cubensis became very popular in cultivation, especially in universities and in closets at universities. I call this Psilocybe cubensis scholarship fund. No one really got rich from this, but they paid their rent. They put the money back into the local economy but many people that I've met subsequently, or physicians and even people of some renown, have confessed to me that they enjoyed the benefits of the Psilocybin cubensis Scholarship Fund, growing psilocybin mushrooms literally under their bed, in their closets, in the dormitories. We sponsored a number of conferences from 1976 to 2017. For 40 years, we did the Myco Media conferences, myself, Jeff Chilton, and Jim Jacobs. And then our final conference was here. I realized that I knew the Merry Pranksters and the Grateful Dead circles, and I also knew Alexander and Sasha Shulgin and Ann Shulgin and Andy Weil. So I had my feet in both communities, and I thought, well, it'd be great. Why don't we bring the Merry Pranksters and the Grateful Dead community in with the scientists like Sasha Shulgin and other scientists who are doing research on this. So in this, there's David Aurora is here, Gary Linkoff. It is numerous other scientific luminaries, and there's Michael Bugh in the very center. So we lost control of that conference, folks. We had 130 people who were tripping on psilocybin, and that's when the group of us decided, we're not doing this anymore. This is the cat's out of the bag, and we can't control it. So up to that point in time, we thought we had pretty good control, emphasizing the academics, the importance of not misidentifying psilocybin mushrooms for basically for health and harm reduction. Other people then picked up these conferences, Jonathan Ott in particular, and the Psychoactivity Conference that you see here in Amsterdam. And there's Albert Hofmann and Sasha Shulgin and myself and Donna Dreyer, Richard Jensen, and other individuals. So, but Terence McKenna, Gary Menzer, Stephen Pollack, and Andy Weil, these are pretty much the main figures in the 70s who were leading the charge on psilocybin mushrooms. So psilocybin mushrooms are interesting in their variability in the production of psilocybin, psilocin, and in this case, baeocystin. So Psilocybe azurescens on the left is the most potent psilocybin mushroom in the world. It's closely related to Psilocybe subaeruginosa, delineated recently genetically, but a very, very close relative. And the psilocybin content, is approaching 2%. Psilocybin dephosphorylates into psilocin. So psilocin crosses the blood-brain barrier. Psilocybin does not. But it turns into psilocin, and that basically this is what's happening in your brain. And then baeocystin levels, because baeocystin was thought to be a toxin. So the analytical methods for baeocystin was commonly used specifically to see if baeocystin levels could be determined in case some of these mushrooms also turned out to be poisonous. Now, two or three of these names dropped off. The one to the very right of azurescens is Psilocybe semilanceata. And semilanceata are liberty caps, packed full of psilocybin, almost no psilocin, substantial amounts of baeocystin. So I'll quickly go through these meta studies. This is from the United States, DSHS, looking at several 100,000 criminals in a survey. If they had one use of psilocybin, there is association, not necessarily causation, but it can be, association with one psilocybin use, a 27% decrease of past year larceny and theft, 22% decreased odds for property crime, and 18% decreased odds for a violent crime. So these meta studies, sociologically, have been sort of the compass that has been pointing to the fact that people who use these psychedelic substances tend to be better citizens. And then a study out of British Columbia here, and Zach Walsh is a major player up here, and found that in terms of partner-to-partner violence, if a individual had taken a psilocybin or LSD trip, there's a statistically significant decreased odds of perpetuating physical violence against their current partner. There is an emotional responsiveness to people who are depressed, enabling patients to reconnect with their emotions and to feel empathy with others. David Nutt and Robin Carr Harris are two of the principal people currently doing research even today, along with Matthew Wall. And then many of you know about this treatment-resistant depression, the results of showing there seems to be a reset therapeutic mechanism that's been proposed. And I know that other speakers in the MAPS Canada series of seminars have talked about the default mode network. I think it's really interesting that from these experiences is an increased nature relatedness and a decreased authoritarian political views. You feel greater empathy towards each other. You feel better connected to the ecosystem, the environment. It's something that people come out of these experiences wanting to do greater good. These have demonstrable benefits to society, reducing crime, increasing empathy, increasing community awareness, and I think raise the level of citizen responsibility in a demonstrable way. There's another example of this, a pro-environmental behavior, also related to engaging with psilocybin mushrooms. So this is one of the most interesting connectograms that I've seen, and that Robin Card Harris put together. It's been widely used, and to his credit. But Nancy Reagan, if you were still alive, this is on the left is a placebo. This is your brain without psilocybin. On the right, this is your brain with psilocybin. Well, obviously, the more connections your brain has, the more ability you have to be creative. And when you're creative, you're happier. When you're happier, you're more creative. So I think psilocybin mushrooms leads to greater creativity and greater happiness. The hippocampal neurogenesis and extinction of fear conditioning is yet another study with mice that were put into cages, and the mice were given a tone. 40 seconds later, they were given an electrical shock to the metal floor. And then after several repetitions, when the mice heard the tone, they cowered in fear. What's interesting about this is that after psilocybin, they were able to dissociate and return to baseline, where the tone dissociated from the expectation of a punishment. And so the extinction of the trace fear conditioning. Thus, we can say psilocybin induces neurogenesis, courage, and kindness. These are leadership skills that lead to better citizenship. So it's proposed in the United States that psilocybin be moved from a Schedule I to Schedule IV. Roland Griffiths from Johns Hopkins is leading the charge on this, with others, Matthew Johnson as well. This is based on purely scientific evidence, not based on politics. They have low or no abuse potential. They're non-addictive. And they have medical applications. Schedule I, typically does not. It has high abuse potential and oftentimes no medical benefit. So these should be reclassified as Schedule IVs. I want to mention quickly another mushroom called the lion's mane mushroom. This mushroom is what I consider to be one of the a smart mushroom. I call it the second smart mushroom, psilocybin mushrooms being the first one. 2 clinical studies on lion's mane mushrooms, on mild cognitive impairment, a double-blind placebo-controlled clinical trial. And these also it found that patients taking these lion's mane mushroom products showed significant increase in scores in cognitive function compared to the control group. Now, there are aranacines that come from the mycelium and hericenones. And it turns out the aranacines are showing the benefit coming from the mycelium far more so than the hericenones. And what is neurogenesis? Well, neurogenesis is the reactivation of the growth of neurons. And it leads to regeneration of myelin on the axons and increased differentiation at the synapses and more intersynaptic crossings and junctions, which allow for new pathways neurologically, for coordination, for memory, for emotive states. So these things definitely are very similar to that of psilocybin. And the progression of Alzheimer's is a function or consequence of that is the formation of amyloid plaques. And Alzheimer's patients, upon resection and autopsy, typically show the production of amyloid plaques and demyelination, the removal of the sheath that's on the axons that transmits neurological signals. And so what is interesting is that numerous studies have shown the resolution of amyloid plaque formation subsequently in mice. I know of no resections of human brains, subsequent to lion's mane ingestion over the long term. But in mice, they've been able to show removal of amyloid plaque, and also behaviorally, seeing that the mice then had better learning ability, and also were able to have better maze solving ability as well. Some of you have seen my other talks on this. I go into this in great detail. But we decided to do some comparisons of lion's mane, mycelium, and fruit bodies with a company called Neurofit in France that does preclinical testing for potential drugs that could help prevent dementia or Alzheimer's using pluripotent stem cells. So there's a control, and the brain-derived nerve growth factor is also a positive control. So one's saline, the other one's a positive control. And the idea is, and these are young neurons, so basically they're growing out, they're extracted, and they're spiked into resting neurons, these pluripotent stem cells that are differentiating into neurons. And then the mycelium grown on rice. So basically you can see the mycelium grown on rice, and this is only over 108% increase in the development in about 7 days in sextuplet. So it's a good number of replicates there. So that was interesting. Great. We found that. We actually found that the fruit bodies depressed and were below the saline solution, actually interfered with neurogenesis. I don't normally talk much about that because I don't want to spread mycophobia, but in this case, the mycelium, did exactly the opposite of what the fruit bodies did. So this is from a 10-day exposure, a little bit hard to see, but this is basically what is being measured, and we use a computer program, analog formula to calculate the number of neurite outbranchings and connections. So we started playing around with legal analogs of psilocybin. So in this case, the brain-derived nerve growth factor over the control, ethyl acetate fractions, and this is 7 days in sextuplet. And lion's mane increased 111%. That was pretty close, 108% before. These three psilocybin analogs we tested, well, they increased at 19%, 29%, 5% over baseline. Well, that's the first evidence that we have seen, that I know of. Anyone out there want to correct me if I'm wrong, I'm happy. I'm factually driven. If you can find references that someone else has done this before us, I'd love to see it. I don't know of any. So we did this to look at the psilocybin analogs. And this is really important when we come to this later. Well, then we decided, well, why don't we stack these? Let's put the lion's mane mycelium. In this case, it would be 108, 112. Now it's 114% with this test. So we have pretty good reproducibility. 3 separate tests, several months apart, several different batches. So, it's fairly consistent. And then we have these psilocybin analogs, the legal analogs, and we chose this one in particular. Now, what's interesting, if they were activating on different neurological receptors, let's just say they were, then you'd have the added cumulative expected effect of 114 and 107. That'd be 121, 122, 122%, 22% increase. We combined them and we found synergism. We actually found it increased neurite outgrowth by 36%. And so we've repeated these experiments, and we're getting more and more interesting result on the entourage effect of not only psilocybin analogs, but psilocybin analogs stacked with lion's mane in a natural form. Erinacines are about, I think, $25,000 for 5 milligrams. So erinacines are not going to be commercially available, folks. The lion's mane mushroom mycelium is easy to grow. Psilocybin mushrooms are easy to grow. Psilocybin molecules, the market rate, I think, is around $5,000 a gram. You can grow psilocybin mushrooms for the price of portobellos, and they contain 1%. So the rush to patent an expensive molecule therapeutically meets the requirements of the FDA for standardization, et cetera, but actually it disservices and does not address the needs of the masses, of individuals who cannot afford. You know, I am a privileged white person. Many of you people, many of us people are also in the privileged white class. Think of the trauma, the poverty of Black lives, ethnic groups that have endured generations of prejudice. I don't walk down the street in fear of the police arresting me. My grandson is half Black. He wears a hoodie. He is a stereotypical target for the police. And I have to encourage him, please don't use your hoodie. He went out and bought an air gun. I said, please don't take that air gun in public. I mean, this is the thing that so few of us understand. The trauma that is being put upon the lower income people in poverty that have experienced generational harm. They need the benefit from these therapeutic substances as well. The natural form gives them access to that because it's economically affordable. The pure chemical form does not. And so I think this is psilocybin is pure molecules for the aristocracy. And I think that we should really address the humanity of making these substances available to all people at all levels of society. So now I want to bring back this to your attention. As it turns out, with this trace fear conditioned response with the mice, something very interesting happened. At the full dose, the high dose of psilocybin, when they then, after the high dose of psilocybin with these mice, when they gave the tone, the mice expected to be shocked in 40 seconds. It took 10 rotations or repetitions of no shock before the mice dissociated that the tone would be followed with a punishment. At low doses of psilocybin, 1/10 the amount, below the threshold equivalent to that of you would feel any effect, any intoxication, it only took 2 rotations. Low doses were more effective potentially than higher doses. It could translate, if this is translational medicine, that low doses of psilocybin could be better for PTSD than high doses. The high dose, of course, you have the therapist there, you're in a hospital environment, you've got medical support teams, et cetera, et cetera. The low doses at sub-threshold levels of intoxication may actually prove to be more beneficial. And I would like to mention that your neurons may grow a little bit in 6 hours, but typically these neurons take days, weeks to proliferate, and to have neurogenic benefits upon cell division, I think will lead to more synaptic junctions, more development of more neurons, and could be more beneficial. So I propose a stacking formula, psilocybin and psilocin, or psilocybin mushrooms at 1% concentration with aranacines and lion's mane. The lion's mane mycelium here could be 200 to 2 grams per dose, so I didn't put that in there. Stacked with nicotinic acid, niacin. The reason being niacin is a vasodilator. With more vasodilation, you have more delivery of these substances to the endpoints of the nervous system. Neuropathies oftentimes present themselves as a deadening, for instance, of the fingertips and the toes. So because the vascular system is better able to deliver these neurogenic compounds. And then thirdly is that those of you who've taken niacin, you get the niacin flush. It's itching. So the idea of stacking niacin with these substances, if someone did try to get high from them by taking 10 to 20 times, 10 to 20 pills, they'd be taking up to 2 grams, up 4 grams of niacin. Well, you get really itchy at about 100 milligrams. And those of you who had a niacin flush, when you take several grams of niacin, it becomes the antabuse of then taking psilocybin to get high. I think this could legally be a pathway for allowing Health Canada or the FDA to understand that because it's stacked with an adversive, it prevents the abuse of it as an intoxicant, but gives you the benefit then of his neurological properties. There's a Fadiman protocol by James Fadiman. There's mine. I talked to James about it. We both just made this up. But the idea is you take it for several days, you stop, and then you clear your receptors and you start it again. This is actually consistent with traditional Chinese medicine and the use of medicinal mushrooms as well. My problem child is Psilocybe azurescens. Psilocybe azurescens causes temporary paralysis. This is very important. We don't know if it's originasin or not. That's putatively one of the compounds that are being suggested because chemically it's more related to toad venom than to psilocybin. But this is just speculation right now. There's something that happens with azurescens and Psilocybe cyanescens and also Psilocybe subaruginosa, all of them grow on wood chips, that causes temporary paralysis. And if you were not expecting this, or if you're camping in the snow and you went for a walk, you could die of hypothermia if you couldn't move. So be forewarned that anyone taking these wood chip psilocybes, so far, these 3 species, this wood chip paralysis, if it is unexpected, could result in panic attacks and a very bad experience. So this is indeed a big concern that many of us have. So I know a number of toxicology centers are tracking this. The North American Mycological Association at NAMYCO.org, NAMYCO.org, has a poison report forms, and they're collecting adverse events with psilocybin mushrooms and Psilocybe azurescens and cyanescens in particular. Here is Shannon, how am I doing on time? I have about 4 or 5 more minutes that I want to conclude on this. But let me share this video with you. It's about 2 minutes long, so it'll give myself a voice break.
Greetings, and meet Psilocybe azurescens. Psilocybe azurescens is probably one of the most potent mushrooms in the world. It contains psilocybin and psilocin up to 2% of its dried mass. Now think of that. 2% of the dried mass of this mushroom are psychoactive crystals. Why would a mushroom produce so much? We don't know, but it certainly has attracted the interest of humans. The bluing that you see here is a bruising reaction. It's indicative of psilocin as it degrades. And the more bluing you see, the more psilocin there once was. Now, psilocybin dephosphorylates into psilocin, and when you ingest these mushrooms, psilocin becomes a serotonin agonist. It means that the psilocybin or the psilocin becomes a temporary neurotransmitter, opening up the floodgates of the senses. Now, this mushroom is sinuous. It's got a sinuous stem, which means it bends back and forth. It's bluing very, very strongly, and it has these very indicative spore color here on the annular zone. And the spore color is purple-brown, and the mushrooms are bluish. Those 2 features in combination pretty much de facto determines its psilocybe. Now look how bodacious the rhizomorphs are at the base of the stem. This is a large psilocybe, by far the largest one that I know that grows on wood chips in the Pacific Northwest. It is now a popular one to have in your backyard. It's just fun. It's just naturalized in the woods here. And this is a beautiful fruiting of them. And the psilocybes, many of the woodland psilocybes have this chestnut brown to caramel color. This one is unique in that it's got these umbos, and there is a broad umbo, and the cap is very circular. The bruising reactions just from impact of rain, perhaps. This mushroom is one of the most fascinating and interesting ones to grow. Those who choose to partake of this mushroom should be warned. These are exceptionally potent, and oftentimes they can cause temporary paralysis, loss of muscle control. So that is not a good thing. It seems that most people who boil this mushroom in hot water, those symptoms seem to be alleviated. But this is clearly a sacred species, and I love just personally touching it. This is not a mushroom that I enjoy eating. It's almost too potent for me, and but it is a species that I greatly admire. I love touching it. I love handling it. I love seeing it, and it's a great indicator of the habitat in my magical mushroom forest that is very conducive to spiritual experiences.
So be forewarned, folks, this is really important. Now, an article that came out by associates of the Usona Institute, Bill Linton's spinoff group. This is really interesting because you're looking at other compounds, baocystin and norpsilocin. I have bioassayed baocystin with no noticeable effect at 10 and 20 milligrams. What's interesting is that these MAO inhibitors, some of which are present within the mushrooms, by the way, but in very, very small amounts, harmine in particular. Well, it's Syrian rue, nutmeg, turmeric, kava, passion flower, and ayahuasca, Banisteriopsis caapi, that is. And some bipolar drugs have MAO inhibitors. So this is what I'm getting at. If you had turmeric tea or a kava, and you took a dose of psilocybin mushrooms, and your friend did not, you could be getting a much more massive psilocybin experience because the MAO inhibitors allow the passing of these other psilocybin analogues, otherwise which are not active, to cross the blood-brain barrier. So the foods that you eat do influence your psilocybin experience, and maybe remarkably so. So be very careful about this. I think the addition of chocolate, Aztecs use chocolate and psilocybin mushrooms. Chocolate is a natural marriage with psilocybin mushrooms. How many other MAO inhibitor foods are there out there heretofore yet not discovered, or when there's a combination of plants that are being used can elicit MAO inhibition? This is yet to be determined. So here is an app that Quantified Citizens developed. It's at microdose.me. It's the largest global microdosing study. It is designed for you to create baseline of your experiences, your cognitive ability, your memory, coordination, and then you basically then report it's anonymized. It's gone through reviewed to be medically acceptable according to the Code of Ethics of Medicine in Canada here. I know they're working on some more additions and improvements of that. But basically, you have a memory test, and then from microdosing or from your experiences, then you can chart your mental acuity over time in terms of hearing, in terms of sight. And so this is something that I encourage people to explore. It is non-commercial. It's designed to create data so it can help researchers preclinically and then clinically. And then coming up very shortly on Monday, June 29th, is this movie Fantastic Fungi that Louis Schwartzberg has put together and has excited people all over the world. Amazing timing and having the movie released. And now you can rent the movie, I guess, on the different platforms for $5. But as a result of the inspiration for the movie, these musicians have come together, and it's a 2-album release coming up June 29th. I get nothing from this. I'm just in love with music and electronica and world beat. And my friend Amani from Desert Dwellers has several songs on here. So I encourage you to tune in to June 29th for the international release of Fantastic Fungi Reimagined. And there's some really, really great music for voyaging and journeying. So I wanted to say thank you all. I wanted to acknowledge the indigenous peoples on whose land most of us are visiting or have migrated to. And we are all connected with our love of fungi. We all migrated out of Africa. We all have customs and traditions. Some of them have been preserved, many of them have been lost, but we certainly can benefit from the wisdom of First Peoples and Indigenous peoples who've had a traditional use of these substances and have been able to use them over the centuries in a responsible way. One of my biggest concerns by far is the irresponsible use of psilocybin mushrooms and other psychoactive substances that could lead to bad publicity or these clinical studies not going forward for the best of societal benefit. With anything that is powerful, there are going to be casualties. That's just the way of science, the way of medicine, the way of nature. But as we evolve, we get smarter and better. I think there's a very strong argument that can be made for natural products standardized to 1 or 2 markers, and that the expense of psilocybin in its pure form, even though it has, in conventional medicine, very well justified rationale, and in the importance of standardization, I do think that can carry over to natural products that also can be standardized to the active constituents of note. And I believe that the entourage effect will give you much better benefit neurologically to make you smarter and nicer. And that's what I think psilocybin mushrooms do. Psilocybin mushrooms make you smarter and nicer. And we need a lot more smarter and nicer people. And at this time, critical in our ecosystem, and I'm involved a lot in COVID research right now, but it's something many of us have been predicting for a long time. When we lose biodiversity, when we cut down the forests, when we have we disrespect nature, we're dismantling the immunological networks of nature. I believe the mycelium is an immunological network of nature that influences not only the food chains, but also influences our emotional and intellectual well-being. And it's time that we reinvest in the mycelial networks that literally exist on every footstep that we walk and we share on this planet. So that's it. Thank you very much, MAPS Canada. And I'll take a few questions, Shannon, if you wish.
Absolutely. Thank you so much, Paul. And that was an amazing presentation and extremely enlightening. So we're super grateful to have you here with us. So we actually have over 23 questions. So I'll give you the top few and we can get a few in here. So the first one has to do with microdosing. Hi, Paul. First of all, your work has completely altered my life. So thank you endlessly for that. Second, regarding microdosing, what is the neurotropic benefit of your suggestion of 5 days on, 2 off versus third wave suggestion of once every couple of days?
Well, it's an educated guess. And for the same reason, the analogy I like to make is I love coffee. I drink coffee only in the morning, typically. But I drink coffee 4 or 5 days. I don't drink coffee for 2 or 3 days. That next cup of coffee I have after a several-day break is like the most potent cup of coffee I've had in months. The idea being that you're washing your receptors, you're resensitizing them. You want your endogenous, pathways to still be intact. You don't want to be addicted to a stimulus that then downregulates your innate ability to be able to regenerate neurons, for instance. We're all on the slippery slope towards dementia. Bad news, folks, we're all going to die. And as we get older and older, we lose intellectual acumen. Mm-hmm. And so I think that microdosing and then letting your receptors clear and then re-stimulate them will then be more provocative in eliciting neurogenesis than just constantly stimulating them with a neurogenic compound. This is supported by many other medicines that, and so you don't normalize and acclimate to a stimulus and lose the innate ability of what you have already within you.
Perfect. Thank you so much. I was looking down at the bottom of the questions, and there was one actually on microdosing lion's mane. Are there any risks associated in taking reishi, chaga, and lion's mane on a daily basis? Is there a benefit to taking breaks from mycelium microdosing for certain species of fungi?
Excellent question. Excellent question. I really don't know the correct answer. I don't know the truth of what the best answer will be. Ostensibly, even with microdosing or even taking mushroom supplements, taking a break, like over the weekend, or taking a several-day break and then begin your regimen again. I take mushroom capsules and mushroom extracts 5 out of 7 days of the week. How many of you actually take your vitamin D every day or your vitamin Bs? I wish I'd be so disciplined to take them every day, but I don't. So I'm naturally interrupting my schedule all the time. I don't have one of those old pill boxes that older people have: Monday, Tuesday, Wednesday, Thursday, Friday. So I'm actually interrupting my schedule. I didn't take didn't take my mushrooms today, so then I take them. So yeah, I think whether it's psilocybin or whether it's medicinal mushrooms or nutraceuticals, I think it's important that you don't normalize your body just to one group of compounds. And chaga and reishi and lion's mane, think of it this way. There's about 2 million species of fungi, about 150,000 species of mushrooms. We've identified 14,000. For millions of years, you can argue, I think very convincingly, primates have used mushrooms. I mean, who's going to argue that primates have not used mushrooms for millions of years? I mean, raise your hand when you have 23 primates already known to identify and eat mushrooms. So using that selection criteria empirically of our primate ancestors and our hominid ancestors and our Homo sapien ancestors, we've been able to go into a library of knowledge. It's like taking one species, one book at a time, and reading it and saying that this thing is helpful. We've selected it down to a group of about 200 species that we know are culinary and medicinal, about 50 species that are very potent in this department, and about 10 to 20 that are superstars. And now chaga is a superstar, reishi is a superstar, lion's mane is a superstar, agarikon is another one, Misima, Phalanus lentius is another one. So there are numerous species in that library of knowledge that through our ancestors, we've been able to select it. The studies that are most interesting, you go to mushroomreferences.com, it shows the mushroom mycelium of turkey tail is a prebiotic for the microbiome. Since about 80% of your immune response comes from your gut. The fact that turkey tail mycelium, not the fruit flies, the mycelium, shows as a prebiotic for the microbiome, enhancing Bifidobacterium lactobacillus and inhibiting Clostridium staph and E. coli. This is the amoxicillin-treated patients where the E. coli proliferates out of control. And so looking at a randomized placebo-controlled study, look it up at mushroomreferences.com, these mushrooms are helpful for the microbiome. When you have a healthy microbiome, you're able to digest more nutrients from other foods. And so I think our ancestors have been able to select out these mushrooms as prebiotics, helping probiotically in your microbiome, the microflora that's important for your sustained health.
Wow, thank you so much. Speaking of agarikon, aren't you right now on the search for agarikon to unmask the largest library in the world? Do you want to speak a little bit about that?
Yeah, we have 65 strains of agarikon right now. I created Agarikon Press in 1981. It gives you an idea how long I've been focused on agarikon. And we have the largest library in the world. I'm bound by several confidentiality agreements, but I can say, because this Harvard professor announced it, so I'm not breaking confidentiality here, Harvard. I'm just repeating what this professor said. We both spoke at Singularity University Exponential Medicine. I referenced people to that talk in November of 2019. And we sent blinded samples to them of mycelium. And literally his response, and this is before COVID, we did this in December. He is testing anti-inflammatories because he's looking for anti-inflammatory agents that can stop Alzheimer's progression. And Alzheimer is a consequence of it is neuroinflammation, or a precursor cause of it is neuroinflammation, two sides of the same coin. So he then got the test results back, and in his words, great scientist, what is this sample? It dropped the cytokines like flies. Never seen anything ever do this. He's done fruit body mushroom extracts and all those things. Never seen anything that has done anything like this. There's no cytotoxicity and extremely anti-inflammatory. And we revealed to him it was agarikon.
Wow.
So I'm in the search for the super strain of agarikon. Mary Hunt, who doesn't get enough credit, in medical history. Mary Hunt was a lab assistant who in 1943 went and found a moldy cantaloupe in Chicago that led to a super strain of Penicillium chrysogenum, the penicillium mold that produces penicillin. Mm-hmm. Alexander Fleming discovered it in 1928, but it could never be commercialized because the strains wouldn't produce enough penicillin. Her strain, her moldy cantaloupe, led to the industrialization of penicillin that many historians believe was a major factor of winning World War II. Because the Japanese and Germans did not have penicillin. They did not have good antibiotics. If you wound a soldier, it's 8 to 10 people to take care of that soldier. And then they could die. So it stresses the entire societal infrastructure. If you have an antibiotic, that soldier can be healed and then go back into service. So her strain was a super strain. If Harvard Medical School, this very small set that we sent them, found such activity. Well, I have 65 strains of agarikon now, the largest cultural library in the world. So we're writing a monograph. I've got 3 PhDs on staff. We're writing 2 peer-reviewed articles per year in the scientific literature. If people want to look at the multivariant effect on the interleukin responses, go to the 2 recent papers that we just published, one in the Journal of Inflammation on mycelium, and you can see the results of this. But again, this is like fine-tuning to find the right natural product that can elicit an immune response, which is typically pro-inflammatory, but we're able to invoke interleukin-10s and 1RAs, which are anti-inflammatory cytokines. So having those stacked together from a natural product is a great benefit.
Wow, that's absolutely amazing. And
I'm talking so much. I'm so crying.
I think there's a lot of people who are going to support you in this, because that's just amazing, amazing findings there. In regards to psilocybin, if you had to guess, when will psilocybin be legal in Canada?
Canada is way ahead, especially with end of life. Mm-hmm. We're all going to die. What government is going to try to control my consciousness the last few days of my life? What are you going to do, arrest me? When psilocybin allows you to meet your death with dignity and kindness and with courage, I experienced several relatives of mine who have died, one of whom died in fear, my father. He asked me for psilocybin. I turned him down. I didn't have the courage to give him psilocybin. And he died in fear. So what Canada has done is, the leaders in the Canadian psilocybin movement for end of life have made a very powerful argument that you have the right at the end of your life to take psilocybin, because we know it's therapeutically beneficial. We know from so many reports and now case studies and now clinical studies that people at the end of their life facing cancer and other diseases are better able to reconcile. And see, when it's like, if God forbid, if you have a brother or a relative who becomes a murderer, goes to prison, well, the whole family's affected. When you have a person dying in stress and fear and anger, the whole family's affected. That trauma is a shared trauma. Now, let's reverse that. And there's so many cases of this, and many of them have been videotaped, and they're part of films that have come out and are coming out, of the person who is dying becomes a therapist to the family, telling them it's okay. What's the societal benefit of that? It's huge. And so I think it's time for us to take these medicines seriously and to realize that they have societal benefit. The concern I have is the monopolization of psilocybin as a therapeutic molecule. I think this is something that everybody should be concerned about. We need to make sure that the custodians of this knowledge truly have the commons at heart. I'm a full believer in capitalism. You have to be able to make the investment to have the vehicle to get it to market. People want to open source everything. Great. Who's going to risk their life savings, their house, everything to get something to market unless they have some type of intellectual property? All patents after 17 years are given to the commons. So patents are a stepping stone in innovation for commercialization that ultimately benefits the commons. The problem is that if you have a group of investors that invest in one molecule like psilocybin, and they see a natural product company that wants to democratize this and offer a competitive treatment at 1/100th the price, that is a threat to their investment. And these very seemingly nice people are going to take off their mask and reveal their true characters when they aggressively start going after everyone else who they see a threat to their investments. This is something we all should be extremely concerned about.
Absolutely. Yeah, no, absolutely. And we would love to hear more about that. Like what can people do? What can we do as a society in order to change that?
I think the best thing to do is to encourage and invest in academic research and independent institutions, and publish as much as possible, get as much information out in the commons as one can. If people are going to go to the patent path, then I think they should, and they go to the commercial path, they should have a dedication to the commons. Nature should have a seat in the board of directors. That's what we lack. We lack of having the commons and nature having a seat at the table. And it's time for us to elevate nature to its rightful place, because ultimately the destiny of our ancestors, or the destiny of our progeny and generations to come, is dependent upon the activities that we have today. So I would encourage people to contribute to MAPS Canada. People can contribute to all the institutions that I showed that are involved in research. And I think people should just reach out to those people who are kind of going off the rail here and say, we understand you're young, you're highly motivated, you want to make a mark, but just bring them back on track. I think, the lessons of psilocybin from the psilocybin experience is to understand that, we are all part of this giant oneness. And I think it's important that we pay attention to the voices that we hear from the mushroom experience.
Absolutely. One thing that I really liked that you said in another talk that you did was, we hear the voices of our ancestors through the psilocybin mushrooms, asking us to step up with courage and kindness to be able to protect the ecosystem. And we need to do this together. Did you want to talk a little bit more to that? I find that's so important.
Well, that was really well said.
Well, you said it.
Yeah, do it better. I think the meaning is obvious, and I'm not immune from criticism. I have learned lessons over the years. I'm very grateful for my position now, but I feel an inherent responsibility. It's a legacy that you leave, folks. It's not the money that you make. Mm-hmm. How are you going to be remembered? Are your descendants going to be proud that they're related to you? Or do they want to be not associated with your memory at all? So this is why the concept of 7 generations that First Peoples and Indigenous peoples espouse, they know an innate truth about the universe and about life. And I think the industrialization, largely led by the European in general, and by our society today, and many of the other commercial enterprises, is hijacking, the return on investment. When the return on investment is microseconds with computers that are located as close to Wall Street as possible, because 2 blocks closer to the New York Stock Exchange can give you a microsecond better advantage in fact trading. That's not helping anybody. No. It's helping the greedy people who can get to the money faster. What's the sustainability that we're facing today? We have COVID. COVID is a collision of ecosystems. the human ecosystem versus the natural ecosystem. And when we deforest and we do factory farming, we're creating recipes for a disaster. COVID is not the only virus that's going to come on the near event horizon. There'll be multiple viruses. And when you have bird flu or swine flu, you have all these viruses co-occurring. As we become immunologically stressed, our immune systems decline, we have greater inflammation, we have greater susceptibility to co-infections. And so it's not just COVID, folks. It's going to be a COVID plus. And the COVID plus, as Jane Goodall recently just said, factory farming could spell the extinction of the human species. Jane Goodall is one smart person. And I think we should take her words to heart. So,
absolutely.
I think we can invent ourselves out of this. And this is why I think psilocybin molecules and psilocybin mushrooms may result in the paradigm shift for a new ecology of consciousness, where we can invent the tools and restructure our society so we can navigate as a community to a better future. So, in my personal opinion.
Well, we appreciate that. Thank you so much. We are over time. Did you want to take another question here? Or do you want to give us some final words?
Well, I want to just say that we right now are all talking. I think most of us, I think there's 245 people right now. There may be a few of you that are journeying right now, so this doesn't really apply to you. But for those of us right now in this, quote, restricted state of consciousness, we see such a small sliver of reality. And when we take these sacred substances, we suddenly understand that there is a universal consciousness. There is a chorus of voices of everything, organisms, minerals, the water, everything is a chorus of voices that are unified in trying to send us a message. We have an inherent responsibility as the quote unquote leaders of this ship that's sinking. We're not only taking ourselves down, but we're taking down the ecosystems. The ecosystems will revolt against us and are revolting against us due to our irresponsibility. Those of us who hear these lessons, I think, can affect positive change. And so my message to everybody is to be kind, be nice. You know, you don't have to react. You don't have to, as my friend Pam says, you don't have to be in their circus. You know, take a higher road. Be compassionate and kind, be courageous, be smart, you know, and encourage other people to follow the same path. We're all in this earthship together. You're either on the bus or off the bus, right? So let's be on the same bus. Thank you.
Thank you, Paul. That was just absolutely amazing. We are more than grateful to have you here on the Maps Canada webinar series, and I can't think of a better message. You know, be kind. So, thank you again for having us here, and we appreciate it, and we hope to have you back again sometime soon.
There's one other thing I want to say.
Yeah, absolutely.
It's really important that we have diversity at these conferences and on these forums. So I want to ask everyone out there who is leading these workshops to have Indigenous voices represented. Invite them. They may say no, but they'll be respected and thankful that you invited them. But let's have a biodiversity of voices. So it's not just white men that are carrying this torch. It's time for the white men to step aside and to give empowerment, and with our extra influence, our extra assets, it's time for us to support the other voices that can lead to biodiversity, because biodiversity, I believe, is biosecurity. Let's invest in biosecurity by investing in biodiversity. And that comes to even speakers at conferences. So that's all I want to say. Okay.
Very good comments there. I'm not sure if you see in the chat, but you have so much love, so many thanks to you. And we have messages from all over the world, Brazil, Colorado, California, all over Canada. So so much love to you from all over the world here, and appreciation for being here with us. So thanks again, Paul.
Thank you. All right. Be well, everybody. Thanks. Okay. Sorry for crying, but that's what happens. Okay. All right. Bye-bye.
So in closing here, I'd like to take some time to thank Paul Stamets for being here on the MAPS Canada webinar series. We would also like to thank our sponsors for helping us out with the webinar series. We have Psychedelics Today, Third Wave, Insight Foundation, Cosmic Sister, Medicine Hunter, Field Trip, The Flying Sage, Canadian Psychedelic Association, Van Lefan, Chakruna, Report on Psychedelics, Unity Herbals, Nested Heart Counseling, Omid Pakman, iNod, Banyan Books and Sound, Conscious Living Network, Cybin, The Wave, and Dosed. So thank you to MAPS Canada volunteers who are assisting with this event. And very importantly, thank you to all of you for being here with us to support the series and to support this important research. So up and coming, we have our next episode, which will be on June 30th with MAPS founder Rick Doblin. Make sure and submit your questions in advance, and our full lineup can be viewed on the webinar page at mapscanada.org/webinar. So thanks again to Paul Stamets for joining us here today. That was absolutely amazing. We are beyond grateful, and we will see you on the next episode. Thanks and have a great day.